When cyanobacteria blooms form, entire bodies of water can be covered by their extreme growth. Neurotoxins are associated with these blooms. However, only a portion of cyanobacteria blooms produce these neurotoxins. The most common of these are called microcystins. There have been very few cases of severe human health effects related to Cyanobacteria microcystins. However, they have the potential to be a potent liver toxin and human carcinogen. Microcystins have been linked to mortality of pets, livestock, and other forms of wildlife that use the affected body of water with persisting cases of blooms. As part of our Be in the Cyan Know project, we have opted to test for microcystins weekly in the Hammond River watershed. First, we collected 1 to 2 milliliters of water into the sample vial. Next, using the graduated pipette provided, we transferred 1 milliliter of sample to the lysis vial containing the dried lysis reagent. We then capped the vial and shook for 2 minutes. Afterwards, we let rest for 8 minutes. Using the forceps provided, we added 1 reagent paper to the lysis vial. We then capped the vial and shook for 2 minutes, and afterwards let rest for 8 minutes. We next used a pipette to transfer the sample into the conical tube. We then closed the conical tube and shook it for 30 seconds. Next, we inserted the test strip into the conical tube with the arrow pointing down. We then let it incubate for 10 minutes. Afterwards, we removed the test strip and laid it flat down and allowed it to develop for 5 more minutes. Next, we interpreted our results and found that there was no microcystins present in this sample. These test strips tell us if microcystins are present or absent. If we were to find positive levels, we would take an additional sample to send to the lab to receive quantitative results.